Malaria is a deadly disease that kills over 500,000 people every year, mostly children under the age of 5 in sub-Saharan Africa. It's carried by the Plasmodium parasite, introduced to the human bloodstream by infected mosquitoes. Usually, malaria is treated with a drug called artemisinin. However, strains of malaria resistant to this drug have been recently found in 5 Southeast Asian countries. Consequently, we need to find new treatments that can be made cheaply, administered orally, and are not toxic. In our labs, as part of an open-source drug research program, we synthesized potential new antimalarials from the triazolopyrazine family, with each lab team attaching 2 different organic groups to the molecule. We used the technique of thin-layer chromatography to track the progress of each reaction across the synthesis process. Our group made a previously synthesized molecule as the control, which was important in verifying that the procedures followed were valid and that any unexpected results were in fact properties of the new molecules. We first made the triazolopyrazine core and added the first functional group. Taking our 2 starting molecules, we reacted them in ethanol to create an intermediate molecule, then closed this molecule's second ring by adding a chemical called chloramine T. This reaction produced an orange-yellow solution of the second intermediary product. Isolating this by filtration, we heated and catalytically reacted it with phenyl alcohol, our second organic group, substituting it in for the chlorine atom. This was our final antimalarial compound. Throughout this synthesis process, we used TLC, thin-layer chromatography, to help us to verify whether we had made a new compound and whether our reaction had gone to completion. For example, after our first reaction, we dissolved our 2 starting materials, our reaction product, and a mixture of all 3, into a solvent of 1:1 cyclohexane and ethyl acetate. We dropped each of these mixtures onto an aluminium-backed silica plate before standing the entire plate in more solvent. The solvent moved up the plate via capillary action, taking each dissolved molecule with it, and the distance which each molecule moved off the baseline depended on its polarity and solubility in the chosen solvent. The first time we performed TLC, we found that our D drop looked similar to the C drop, meaning that our reaction hadn't proceeded to completion, as the spots representing our reactants A and B were also found in C, which should have just been our product. After further heating, the TLC showed that none of the reactant spots appeared in C, and so we concluded that our reaction had proceeded to conclusion. Another more precise analytic technique, NMR, was used at the end of our process to check whether we had made our intended products. After a final group TLC to confirm that each lab pair had made the same molecule, all groups combined their samples to purify the product, first using a separation funnel workup technique, and finally driving off all solvent using a rotary evaporator. Now our product was ready to go and sent off to test against the Plasmodium parasite.